Welcome, everyone, and thank you for your registration and time with us today. I'm Wendy Shelton from Entellus, and I'm going to introduce you to our company and today's speaker, Jason Chan. Why is drug development so hard? Studies show that among phase 2 failures, at least 40% of teams don't understand why their drugs failed. If we understood how a therapeutic candidate works mechanistically, if we understood how different individuals would respond to that mechanism, maybe we would understand better and improve those numbers. Is the era of safe drugs that work in most of the population over? Individual patient responses vary. It is worth pointing out that none of the unobservable physiology and only a small part of the immense individual physiological variability can be addressed through conventional clinical trials. Meanwhile, standards for safety continue to be more rigorous. Physiological modeling and simulation is a powerful tool to better understand how drug candidates work and how each individual will respond, making therapeutic R&D programs faster to execute from discovery to clinic, cheaper, to not pursue inevitable failures, and more likely to deliver improved health and commercial value, doing good and supporting further investigation. Entellus is the largest company dedicated to physiological mechanistic modeling and simulation. Our teams comprise multidisciplinary scientific engineering and software development experts, perfecting our method for over 15 years. Our reusable Physiolab platforms enable timely, efficient modeling of physiological complexity, incorporating data on all relevant biological processes at the cellular and clinical systems biology level. Physiolab platforms are computer models that represent both the physiology of your target mechanism and also how different individuals will respond to that mechanism. Dr. Chan will present case studies to this effect, testing the full range of what's feasible in best case and worst case scenarios, generating testable predictions for experimental endpoints and biomarkers, identifying key pathways that can be linked to molecular targets and genetic variability. Without further ado, here is Dr. Jason Chan, Associate Director of Inflammation and Immunology at Entellus.
Okay, thank you, Wendy, and thanks again, everybody, for joining us today. Presently, you are all on mute. If you have any questions, feel free to enter them via the chat, and we will have time for question and answers after the presentation. So for today's webinar, agenda is as follows. First, I'd like to give you an overview of our rheumatoid arthritis Physiolab platform, discuss how we implement therapies within the platform, and then very importantly, go over the concept of virtual patients and virtual populations. I will then describe the workflow that we have used to identify candidate biomarkers. So the Rheumatoid Arthritis Physiolab was developed over the last 10 years with input from key opinion leaders in rheumatoid arthritis, as indicated by the list of academic consultants on the left-hand side of this slide. In this table, I'm highlighting some recent publications and presentations using the RA Physiolab platform. And I'd like to point out in particular the top paper, which was published in 2011 in Arthritis and Rheumatism, in which our original development partner used an earlier version of the platform to identify a biomarker that was subsequently validated with clinical data from the BEST trial. I'd also like to point out the bottom entry here, which was a poster that we presented at the American College of Rheumatology in 2007, in which we made predictions to explain the effect that rituximab was having on bone erosion. And very interestingly and satisfyingly for us, our predictions were validated in a paper that was published in the Annals of Rheumatic Disease in 2012, and indeed many of the findings in this paper were consistent with what the model had predicted. So at this point, I'd like to discuss very briefly the approach that we use to model disease at Entellus. And here we use what we call the top-down approach. And by that I mean we focus in on the clinical outcomes that one would measure in a trial. So for example, in rheumatoid arthritis, we focus in on outputs such as the ACR score. We next identify the key underlying biology that's required to represent those clinical outputs. So for example, in the ACR score, it's been shown by various studies that reductions in disease activity are associated with this reductions in the synovial cell infiltrate. And so thus we model the various cell types that one finds in synovial tissue and their relevant dynamics. At a layer below, we then model the various key regulatory mediators and cellular interactions that occur within the synovial tissue. And indeed, this forms essentially an underlying chronic inflammatory milieu. So depending on how one perturbs this underlying milieu, this essentially forms the basis of how we implement therapies within the platform. The other key concept I'd like to raise at this point, which I'll discuss more, is the idea of virtual patients. And so depending on how one parameterizes the model, we can represent various patients. And this is important because it enables us to represent both clinical and biological variability. So on this slide, I'm illustrating the physiological and biological scope of the platform. So the RA Physiolab platform basically integrates and synthesizes past and present research to model the dynamic interplay of key cell types as well as an array of mediators in the inflamed joint. Within this platform, we can represent various patient phenotypes. So quite importantly, of course, we can look at patients that have inadequate response to methotrexate, as well as inadequate responses to TNFs, and indeed patients that have variable responses across other approved therapies. And this is very important as we are looking in an increasingly crowded RA field for us to be able to stratify or differentiate any potential new target from the standards of care. Within the model, we represent various cell types that we see in the joint. As I indicated previously, this includes osteoclasts and chondrocytes, as well as various immune cells, including macrophages, T cells, various subsets of T cells, in fact, B cells, and plasma cells. The clinical outputs that we represent include the ACR score, but as well DAS28-CRP. We also look at structural outcomes, including joint space narrowing and bone erosion.